Hi, my name is Songyun Hai. I'm a professor at Ohio State University. In this video, I'm going to share with you our paper that links ATR3, a stress-inducible gene, to inflammation and breast cancer metastases. Many people contribute to this work, and the first author, Chris Wolford, was instrumental to the progression of this project. During my presentation, I will identify our collaborators for their contribution. ATR3 stands for activating transcription factor 3. Overwhelming evidence indicate that the expression of ATR3 gene is induced by many stress signals. Previously, we showed that ATR3 in breast cancer epithelial cells is functionally important. The question then is whether ATR3 is clinically relevant. We examined ATR3 in human tumor samples by immunohistochemistry. As you can see here, ATR3 is expressed in the cancer epithelial cells, which are also positive for pankeratin. However, we also saw ATR3 in non-cancer cells. In the beginning, we thought they were just non-specific background signals, but after month-and-month troubleshooting and control experiments, we concluded that they are real signals and decided to score them. We analyzed tumor microarray containing about 290 patient samples. To our dismay, ATR3 in cancer epithelial cells does not correlate with worse outcome. However, we were very excited to find that ATR3 in mononuclear cells is an independent predictor for breast cancer-related death. We knew that they were mononuclear cells because our collaborator, Dr. Leon, is a pathologist. He scored the signal and identify the cells by morphology. I'd also like to thank Drs. Sutherland, O'Toole, and Millar for providing TMA outcome data and statistical analysis. In hindsight, it makes sense that ATR3 in breast cancer epithelial cells does not correlate with clinical outcome. This is because ATR3 would be induced by various stress signals in cancer cells during their development. Our data that ATR3 in mononuclear cells correlates with worse outcome led us to study ATR3 in the host, that is, the non-cancer cells. We injected cancer cells into either the wild type or knockout mice and examined primary tumor formation and lung metastases. As you can see here, primary tumor formation is the same. However, the knockout mice had much reduced lung metastases. We repeated experiment using different strain of mice, different cancer cells, and different injection models, and observed the same results. Therefore, ATR3 in the host promotes breast cancer metastases. 3 lines of evidence indicate that macrophage myeloid cell is the key cell type for ATR3 function. Using conditional knockout mice and ABAC experiments, we showed that ATR3 is necessary and sufficient for myeloid cells to enhance cancer metastases. Using co-culture experiments, we showed that ATR3 in macrophage is important for their ability to enhance cancer sphere to form invasive structure. We also showed that ATR3 in macrophage is important for their ability to enhance cancer cells to migrate through endothelial cells. We also carried out gene profiling using tumor-associated macrophages. We found that ATR3 promotes macrophage toward M2 skew and also regulates a lot of genes involved in cell-cell communication. We also identified an ATR3 TAM signature that can distinguish tumor stroma from adjacent stroma. This signature can also segregate patients into low and high-risk groups in 2 data sets, the NKI and the Stockholm data sets. I'd like to thank Drs. Ostrowski and Merchant for carrying out the bioinformatics analysis. A significance of this finding is that the gene signature was derived from mouse macrophages, yet they correlate with the human outcome, lending credence to the mouse models we used. So we started our project by analyzing human tumor microarray for clinical correlation that led us to study ATR3 in the host using mouse model which led us back to analyze clinical correlation. The difference is that in here, we analyzed ATR3 gene expression itself, and here we analyzed ATR3 gene signature, that is the downstream genes of ATR3. I'd like to thank our collaborator, Dr. Shapiro, a breast oncologist, for providing clinical insight to us. In conclusion, ATR3 is functionally important and clinically relevant in breast cancer progression. Because it is a stress-inducible gene, our finding has important implication. As an example, ATR3 is induced by all the stress signals listed here. One question we're asking then is whether ATR3 plays a role to promote cancer development under those stress conditions. With this, I'd like to thank you for watching this video. Please feel free to contact me if you have any comments.